Hi, this is my culminating on anthrax, and here's a quick overview of what anthrax is, just to refresh your mind. And here is also a quick introduction about the things we will be going through on this presentation. Unit 1 is biochemistry in relation to anthrax. So we're going to look at the properties of the bacteria. So it is called Bacillus anthracis, and it is a gram-positive rod-shaped bacterium. What is a gram-shaped positive bacterium? Well, it has a thick peptidoglycan, sorry for pronunciation, layer and no outer lipid membrane. It is easier to destroy than a gram-negative bacteria, but it still causes problems. The thick porous layer in the cell wall of the gram-positive bacteria gives greater access to antibiotics, allowing them to move more easily and penetrate the cell and or interact with the peptidoglycan itself. It's also a multicellular bacteria. So we're going to look at the next slide, Bacillus anthracis and cells. So we're going to look at spores, which is a specialized cell type. Spores are a highly infectious form of this bacteria. Upon entrance into the host, specific signals facilitate germination into metabolically active replicating organisms, resulting in a disease pathogens. An infectious particle also is smaller than a virus, but it contains no nucleic acids, neither DNA nor RNA. What happens when the bacteria enters the host cell? The spores activate in a host body and multiply, releasing three proteins: the protective antigen, and edema factor, and the lethal factor. The protective antigen binds to the host cell, allowing the other two protein toxins to enter and kill the cell. So the bacteria needs to be activated in the host cell in order to be able to kill it, and this is a process called germination, where it is an irreversible conversion of a dormant bacteria and a spore into a metabolically active cell. First consists of the reversible activation of the spore by heat or other agents, and the second consists of the irreversible germination process, which can be induced only in the activated spore. So Unit 2 is metabolic processes in relation to anthrax. So we're going to look at the effects of the anthrax toxin. So the toxin can be produced in both a vitro and vivo environment, which means in a controlled and an uncontrolled environment, both of which causes death of animals along with pulmonary edema, accompanied with pulmonary edema. The effects of the anthrax toxin can cause damage to the lung tissue, blood vessels, and alveoli. The lungs can become heavy, purple, and firm. The blood vessels are can become severely congested, and official alveoli contain small collection of extravasated erythrocytes, as well as small amounts of edema fluid. In the images in the screen, you can see visual examples of the damages that can occur because of the toxin. The biggest effect that the anthrax toxin is on oxidative metabolism, which is key for the body, as it is the biological process to convert toxic chemicals into benign ones, such that when insufficient oxygen is available, these toxins accumulate within tissues and cause potential damage. Thus, if there's damage to the lungs, insufficient oxygen is there for oxidative metabolism to occur, which can cause organs to fail and many other problems. Also, to mention, in experiment and in animals, it was shown that there's a 30% reduction in overall oxidative metabolism. So Unit 3, we're going to look at molecular genetics in relation to anthrax. So we're going to look at how sequencing of the anthrax genome or DNA can help save us and help us. So we're quickly going to look at what genome or DNA sequencing is in general. So it's basically determining the order of the 4 chemical building blocks bases which make up the DNA molecule. This helps scientists to know the genetic information which is carried in a particular DNA segment, which allows researchers to identify changes in genes associated with diseases, phenotypes, and identify potential drug targets. Let's look at anthrax in relation to DNA sequencing or genome sequencing. So it allows us to obtain information about anthrax and the deadly illness to help us prevent and treat it. It is a biological weapon, so people who are unfortunately attacked by it, we can help them be treated and survive. By unraveling the bugs, basic biology researchers may be able to devise new screening protocols and decontamination strategies. A team deciphered the genetic code for 2 different types of anthrax, and by comparing them, they were able to see a difference of just 11 letters that separates the two, which can help identify ways to treat each of them. And one of them is the deadliest version. And they identified a multitude of genes that enable the bacteria to cause the disease. This includes sequences for spore survival and activation, and by this information, new drugs could be designed to target the proteins that the genes code for. Unit 4, homeostasis and its relation to anthrax. So research has shown that a key hallmark on this type of toxicity disrupts important cellular barriers needed to maintain homeostasis. And on the right side of the screen, we can see pictures of homeostasis to help us visualize while we are going through the presentation. So what is a central hallmark? Well, it's a distinguishing characteristic and or feature of the disease, or in this case. So a quick example relating to anthrax, well, one type of anthrax is that for skin anthrax, one of the hallmarks is our carbuncle, a cluster of boils that ulcerates. Typically, the carbuncle has a hard block center surrounded by bright red inflammation. This dramatic appearance accounts for its name, anthrax, the Greek word for coal, a burning coal. Just some fun facts. So what is a barrier? Well, a biological barrier are living organisms that help protect the body from pathogens. The psychological function of these biological barriers is to diverse among tissues and respond to the particular needs of each organ, including the supply of nutrients, the absorption of ions, the secretion of waste, and the protection against toxins. These are just a few of their jobs. Some examples of barriers include endothelial barriers and epithelial barriers. Let's look at the direct effects on homeostasis. So let's look at host barriers first. So host barriers provide both physical and immune defense against microbial infection. So let's look at the 3 definitions, which one we went through the host barrier, and then the lethal toxin, which is a combination of a lethal factor and a protective antigen. And then let's look at psychological homeostasis, which is a tendency of the body to maintain important psychological parameters. Now that we've looked at these 3 definitions, let's look at how they go together and showcase the effects of anthrax on homeostasis. So the effects of the LT, or lethal toxin, on host barrier function not only contributes to the spread of the infection, but also disrupts psychological homeostasis. And this is a big deal because psychological homeostasis is the tendency for the body to maintain critical psychological parameters, which include blood glucose level, blood pressure, core body temperature, and a plethora of other things. And if we aren't able to maintain these things, then our body can shut down and soak in our organs. For Unit 5, we're going to look at population dynamics in relation to anthrax and the population that factors that played into the outbreaks of anthrax. So we're going to look at a quick history of the outbreaks, and Canada's not included in here because their contribution was so small that it's not even significant. So the estimated origins of anthrax go all the way back to 700 BC, Egypt and Mesopotamia. But we're going to jump a little bit forward and look at their history from 1979. The first documented clinical case and description of anthrax was in 1752. So the first one we're going to start with is 1979 in the Soviet Union, which was an anthrax attack, and there were 105 victims, but the statistics of how many people survived and how many people died are unknown. In September 2001, on the 18th, in the US, and there were 5 deaths and 17 infected, and this was a case of inhalation anthrax. And then in October 2014, in an Indian village, there was an outbreak which caused 7 deaths. And then the next one was in July 2016 in Russia, and there was an outbreak, and 100 people were infected, and there was 2,300 deaths of animals. And then in June 2018, there was a lot of affected cattle in France, but the statistics aren't known. The last known human case of anthrax was associated with livestock exposure in US 2002 in South Dakota, and there was an increase of animal cases of anthrax in 2019. This is a quick visual to look at the outbreaks in the world during the specified time, just so you can see where there were more outbreaks and where there weren't, just to see how population factors play into this. Let's look at some outbreak factors. So the vaccine, there is a vaccine and it's approved by the FDA, but it's just a preventative measure. So only people who actually need it can get it if they choose to, like people who work with livestock and are more vulnerable to getting anthrax. Let's look at an example. Why were there more deaths in India than there were in the US? So let's see some population factors. So the population density, obviously in a village in India, it is much more dense than it is in US. Therefore, the living conditions are much different, and people are closely tight-knit together, so the transmission is easier. And the living conditions are different because in a village in India where poverty is high, healthcare isn't as good, so there wasn't treatment for those people, so more people ended up dying, whereas in the US there is treatment and people were able to access that. Let's just quickly review and summarize what we went through in this presentation. So we learned how anthrax relates to the 5 units, we learned in this course. So we looked at biochemistry and the properties of bacteria and how they adhere to cells, and we also learned metabolic processes and how the anthrax toxin affects a lot of the body, specifically metabolic processes and metabolism. And then we looked at molecular genetics and how sequencing the anthrax genome or the DNA can potentially help us prevent more deaths to occur in the future from this infectious disease. We looked at Unit 4, homeostasis, and the substantial effects anthrax has on the body, which impacts homeostasis, and specifically psychological homeostasis. And then lastly, we looked at population dynamics and the outbreaks that occurred in multiple parts of the world, and what factors played into how many people were infected, how many people died, and the origins. And we also looked at a quick history of the outbreaks that occurred. Thank you, and that's the end of my presentation, and then the works editor at the end.